Thank you, Asaf. As always, we'll take questions after all of the speakers have finished. I'd like to invite Luciana Borio up next. Luciana is the acting chief scientist at the FDA, and she's also a member of the Forum on Microbial Threats, our second forum speaker of the day today. And Luciana is going to be giving you some case studies of big data analytics at work in the FDA.
Yeah. So just click. Yes. To the left, okay. So good afternoon, everybody, and thank you for the opportunity to be here today and to participate in this topic that I think know less than every other speaker here today. But I'll take the opportunity to use the podium to let you know that sadly my current director of Office of Health Informatics is leaving the FDA very soon. He's here today. But we are going to be hiring. So if you're interested in working on big data, having a big impact, and making a small salary, the FDA might be the right place for you, and please email me. So I was asked to present some of the case studies focused on infectious diseases. And FDA has a very, very broad mission, very broad, as you know. And we rely on data every day. It's the soul of FDA is data. Every decision that we make is grounded in data. So it's all about the data. And we rely on big and small data. They're both integral to our daily operations. And I don't think that we have even really think about big data, small data, unless you're really responsible for building those architectures that can accommodate the volume of data. And generally speaking, the more data we have, the better, but one caveat is that all the data that we consider has to meet some standards for quality and integrity so that it yields reliable and interpretable data. So it's not just the quantity, but also the quality. And we have several big initiatives underway. There are so many, and I really struggle to just pick a few that I think would show how we are leveraging big data to further our mission. And I focus on infectious diseases mostly, but when I walked in here this morning, Lonnie asked me about a program that we have in compliance, and I had not included it, but I promised to just say a word or two about it, and it's called PREDICT. It's for prediction of risk-based evaluation for dynamic import compliance targeting. So FDA has more than 30,000 import entry lines or distinct products that comes across our borders. And this program is relying on automated data mining and pattern discovery, on open source intelligence, and relies also on automated queries of FDA databases to inform compliance actions. And the result is that we now have a score for each entry line on the basis of these risk factors that are mined. It has allowed us to really tremendously increase the number of the green light decisions, so may proceed immediately at the border, and then allows us to target those more limited resources, compliance resources, to evaluate the highest risk lines. And importantly, the reviewers now are giving a score and reasons why the product may not proceed immediately. So we can see how it's been really important not only to make informed decisions about compliance at the border, but also allows us to focus more limited resources in areas that may represent higher risk. Now, going back to the infectious disease space, so one area where it has made a tremendous difference has been in the area of food safety. In 2012, we launched Genome Tracker, which is the first open source whole genome sequencing integrated network of state, federal, and commercial partners for detecting and tracing foodborne outbreak pathogens back to their source. This Genome Tracker network has sequenced more than 51,000 isolates and comprises 17 terabytes of data. I have no idea whether this is big or small, but it sounds big to me. And the network is regularly sequencing more than 1,000 isolates each month. So it's been a true paradigm shift in the identification and investigation of foodborne outbreaks, because we rely now on whole genome sequencing, allowing us to trace foodborne pathogens back to their source more quickly and with greater accuracy. In 2014, we conducted an environmental sampling at almond and peanut butter facilities as part of an effort to gather baseline data on the presence of foodborne pathogens in nut butter processing facilities. The samples from one of the facilities tested positive for salmonella, and whole genome sequencing on the salmonella strains from this facility, as well as from a small number, very small number, of clinical isolates collected from individuals who have become ill from salmonellosis in the previous months. And again, they had no clear epidemiological link. Oh, something. I may have clicked. There we go. They had no clear link. They determined that the strains were identical, and confirming that the isolates were related to each other, and that the individuals who became sick represented a cluster of illnesses caused by the same organism linked to the nut butter processing facility. And based on this information, FDA is able to stop the food product from going further into the food supply and stop the outbreak. And you can see that with this technology, it has really shifted very early on to allow us to do disease detection and take control measures. And we work, this is a partnership between FDA, CDC, and others, but predominantly managed by FDA and CDC. Another area where FDA is leveraging big data is using next-generation sequencing to analyze drug resistance in the review of new drug applications for antiviral drugs. So next-gen sequencing allows us to identify lower frequency mutations in viral genomes that might not otherwise be detected using the more traditional Sanger sequencing methods. In one example, our scientists, doing their independent analysis for drug resistance, relying on next-generation sequencing provided by a sponsor of a drug to treat hepatitis C. And this analysis identified that low-frequency treatment emergent substitutions that appear to be associated with resistance, and that although they are very relatively infrequent, had the potential to reduce the effectiveness of the drug and affect also future treatment options. But the result of these analyses, which are now in the label, in the product label, are also used day in and day out by clinicians to inform optimal treatment regimens. And I can tell you this was no easy feat. It was the first time that the Division of Antiviral Products had to build an entire infrastructure to support this type of data analysis. The data set was approximately 1 terabyte, and we had to develop a business processes for transferring and storing the data, as well as the analysis tools and IT solutions to be able to deal with this type of information. Again, essential for FDA as a regulatory agency with the primary responsibility with, that conducts primary data analysis to be able to have this capability that can really analyze across clinical trials, across drug products and all. Next, I'll talk a bit about the role of big data in antibacterial resistance. As the prior speaker from DoD mentioned, clinical laboratories use breakpoints, which is the concentration of an antibacterial drug at which a species of bacteria is susceptible or resistant to a drug, to evaluate susceptibility of bacteria that have been isolated from patients. So breakpoints are used all the time to assess the likelihood of treatment success and inform the care of patients. And also, we also sometimes have to provide special infection control procedures for certain patients to prevent further spread. So the information gathered is very, very critical to patient care. Breakpoints are set and updated by standard setting organizations and the FDA, and they may need to change over time to keep pace with new resistance mechanisms that develop. So we are supporting a project by investigators at Johns Hopkins to create a database with medical information for more than 5,000 patients across 3 different hospitals. These patients are being treated for bacteremia, and the database includes information on the characteristics of the bacteria causing the infection that has been isolated from these patients, the antibacterial drugs that are used to treat the patients, patients' risk factors, and then their outcomes. So this information in this database will allow us to utilize real-world clinical data to establish and update the breakpoints in a very clinically meaningful manner. And lastly, I'll mention one other application in the area of infectious diseases, which is as it relates to clinical trials. So this is an area where big data can make a big difference, is to help us design better and more efficient clinical studies for antibacterial drugs. I won't go into much detail about some of the challenges associated with mining that data, but if you've been using Amazon music services and downloading music, you know that they have this tremendous ability to guess what you like and also know what you bought 10 years ago. Even sometimes they like to forget your prior music tastes. It kind of haunts you, but they just share this data freely. And of course, it's not the same in the healthcare system where the data are more siloed, there are privacy issues, so it's not readily available. But this is an area that I think we're moving towards improving, and because the promise is so tremendous. So I'll go take a step back and remind folks, and I think everybody here knows, that conducting trials for drugs for serious acute infections, it's usually very, very difficult and very expensive. Patients are acutely ill. They must receive treatment urgently. The diagnosis, the specific diagnosis of the microbiologic etiology may take several days. And optimally, of course, patients should have no or limited use of antimicrobial therapy before enrollment in the study, because it's very difficult to evaluate the impact of an investigational drug properly if the patient is already enrolled in the trial but is already getting better by the time that they are enrolled and receiving the investigational therapy. So with FDA support, Duke's Clinical Trials Transformation Initiative, or CITI for short, is conducting this large observational study that identifies risk factors for patients that can go on to develop hospital-acquired and ventilator-associated pneumonia, HABP for short. So these trials are particularly difficult to conduct. Enrollment rates per study site on average are very low because of the challenges I mentioned, and sometimes we need more than 100 sites to be able to complete a study. So they're lengthy and they're expensive and frustrating to sponsors. So the project by CITI is working to identify risk factors associated with the high risk of developing HABP. And then the data can be used to identify patients that might be at high risk, so it's predictive, to develop these conditions. And the idea here is that they can be pre-consented at the time they encounter the healthcare system or before they develop the condition. And then they can be enrolled in the trial at a much earlier time point in the course of their illness. And another benefit for this approach is that it will allow us to adjust the inclusion and exclusion criteria so that we can increase enrollment. We suspect that a lot of patients are being excluded from clinical trials today for unnecessary reasons, and they don't necessarily reflect the diversity of patients that are actually treated in a real-world scenario. So this data is going to help us create more efficient and more robust clinical trials, we hope. And the ability to conduct improved and more efficient trials is really one of the conditions that will be necessary, we think, to support the pipeline of new antibacterial drugs. So I'll stop here and look forward to questions at the end of this panel. Thank you.